wildlife service, and I'll be giving the slides today for you for the webinar. We also have Ellen Denny, our monitoring design coordinator, who will be doing the questions at the end of the webinar. Also, Sarah Schaefer is our web designer and developer, and she'll be helping with technical support, typing in the links for some of the resources that I talk about, and running some of our polls later on in the webinar. And again, we will be recording this webinar and posting it along with the answers to the questions and the slides on the webinar page. So just to make sure that we're all on the same page about phenology, we like to start with our definition. We consider phenology the study of the timing of recurring plant and animal life cycle stages, or Phenophases, and their relationship to environmental conditions. We'll be talking a lot about Phenophases today, so just keep in mind that these are the plant and animal life cycle stages. So phenology is something that you see all around you. It's the science of the seasons, and different examples include blooms and buds, hibernation, migration, emergence of insects. And so it's something that you can see in your everyday life, and it's very easy to observe. Here at the USA National Phenology Network, our primary goal is to create a standardized data set for use in multiple types of research. Our mission is twofold: to make phenology data, models, and related information available, and to encourage people of all ages and backgrounds to observe and record phenology. Nature's Notebook is our plant and animal observation program. And it's something that both scientists and citizen scientists can use to collect phenology information. We just wanted to do a quick question here to find out how many of you are already observers with Nature's Notebook, or whether you're just here to learn more about the program. So we're going to launch our first poll and ask you whether you're already signed up, whether you've submitted observations, or whether you are just learning about the program now. So go ahead and select the answer that best suits you on the screen, and then we'll share the results in just a moment. Okay, so we're going to share the results with you now. And you might see just a bit of a lag depending on your internet connection speed. So we'll wait just a moment and those should show up. Okay, so it looks like about 42% of you are already observing with Nature's Notebook. 27% have an account but haven't submitted observations yet, and 31% have not yet signed up with Nature's Notebook or are not yet observing. Okay, that's great. We have a really good spread across those different categories. So I hope that this webinar will give you background for those of you who haven't yet started observing, and then we'll also give you some more in-depth information for those of you who are already observing with us. What we're going to be focusing on today in this webinar are our Phenophase protocols. So this is an example of one of our data sheets for a deciduous tree, and you'll see that there's a list of questions on the left-hand side, and each of these represents a phenophase, or a life cycle event for this species. So for our Nature's Notebook program, we're really interested in long-term monitoring of the same individual plants over time. So this means you'll be marking plants and going back to them to make continuous observations over a long period of time. And this really allows you to be able to see the changes in your plants, not just throughout the year, but also to compare multiple years and see how things are different from year to year. We really value the no observations that you make. You can see on that data sheet that we have a Y, an N, and a question mark. And that N represents a no, you did not see that phenophase. So when you observe with Nature's Notebook, we're not only interested in when you are seeing things occurring, but also when you're not. This really helps us to pinpoint when that first yes does occur. So if you're monitoring over time, say you're making weekly observations on a particular plant, you'll be reporting no for a particular phenophase until you see it starting, and then start reporting a yes. And that really allows us to pinpoint when that first phenophase started to occur. So you would know if you were making weekly observations and you had a no followed by a yes the next week, that that phenophase started within the last week. So it really provides accurate data for us to know when things are first starting. The timing of how often you observe really depends on what you're observing and the season. So we say weekly on average, but this might be more often, say in the springtime when things are occurring very rapidly, and it might be less often in the winter when things are dormant. And there is that question mark option. We really encourage you, if you're uncertain about an answer to one of these phenophase questions, you're welcome to use the question mark. You can always go back and change your observations after they're entered and update it to either a yes or no if you figure out the answer later on. So what we'll be talking about in the webinar today are the 3 different groups of these phenophase questions. So we'll be talking about leaves, flowers, and fruits. You might be wondering why we care about these phenophases and why we've selected these particular phenophases. We have worked with many different research communities to try to develop these phenophase protocols. So some of the examples of why we care about these particular phenophases, such as the canopy questions, are they're really a big interest to carbon sequestration or food for insects or wildlife. So for example, if we know that a tree greens up very quickly and that the leaves stay on for a really long period of time, that could be more carbon uptake by a tree. And if you're talking about a larger area, this can really be important for looking at the amount of carbon that's sequestered in trees. This also allows researchers that are looking at remotely sensed images to calibrate and see if what they're seeing from these images can be ground truth by the observations that are made by people observing trees on the ground. For the questions about flowers, knowing about peak flower can help with knowing when wildflower viewing might be best. For example, about a week or so ago in Washington, D.C., there was the peak of the cherry blossoms. And so knowing about when that peaks is really important for tourism. So having more information about that can be really useful for city planning and other things. Also, it's important for pollinators. When we know more about when flowers are opening, we can know if these are becoming mismatched with their pollinators or when pollination can be occurring from year to year and whether that changes. It can also be important for allergy season as well, knowing when the pollen is released on a particular plant and whether that's changing from year to year can help predict when allergy season is going to occur and even allow different companies to put more allergy medication on the market and actually use those predictions. Questions about fruiting can be important for things like food for wildlife as well. So for today, as I mentioned, we're going to be talking about leaves, flowers, and then fruits. And most importantly, we have phenophase definitions for each of these questions, and they were written to be taken literally. So we really try to write these definitions to be very detailed to take out the guesswork from these kinds of questions. So we don't want you to try to figure out what you might kind of infer from these definitions. Just read them and take them literally and don't try to make a guess based on what we're saying. So just to give you an example of that, for this picture here, if you were asked the question, do you see open flowers? And perhaps if you didn't know too much about this particular species, you might say that this was indeed an open flower. However, if you read the definition that we provide, it gives some very detailed species-specific information that tells you, if you read the bold text at the bottom, for this species, Cornus florida, ignore the 4 large white bracts and watch for the opening of small flowers in the center of the bracts. So you would actually report a no for open flowers for this particular photo here. So this graphic will give you an idea of how some of our phenophases are actually nested inside of each other. So when you're reporting one phenophase, you might also be reporting another phenophase for a yes to both of these at the same time. So for example, you might start with a basic deciduous plant phenophase where first you would report breaking leaf buds at the beginning of the season. And then once these change to leaves, you would start reporting leaves as well. Now leaves is one of our overlapping phenophases where you would still report leaves while you also report yes answers to other phenophases. So two of the phenophases that are nested within leaves are increasing leaf size and colored leaves. So even when you see the increasing leaf size taking place on the plant, you would also continue to report leaves. And the same for colored leaves. Once you start to see some of those leaves changing to their late season colors, you would start reporting yes to colored leaves, but then continue to report a yes for leaves as well. This is similar for flowers or flower buds, where that phenophase is an overlapping phenophase. And once you start to see the flowers open, you would continue to report a yes for flowers or flower buds. The same thing for fruits, which is the overlapping phenophase. Once you see these fruits turn ripe, then you would continue to report a yes for both fruits and for ripe fruits. And then also for recent fruit or seed drop, you could report a yes for that phenophase in addition to reporting a yes for fruits and ripe fruits. So we just wanted to give you a little bit of botany information that relates to each of these phenophase categories. So to start with buds, these photographs just give you a little bit of an idea of where buds might be located on a stem. So there are two types of buds. You might have a terminal or apical bud that's located at the end of the stem, or you might have an axillary or lateral bud that's located not at the end but more lower on the stem. And you can have both of these occurring on the same stem. And buds will, they can turn into either leaves, stems, or flowers. And sometimes it's a little bit tricky to figure out which of these is going to occur. So we'll have some information on that and how you would report that in Nature's Notebook in a bit. Some buds will have outer scales or protective coverings, especially buds that are occurring on woody plants in colder climates. But they don't always have these scales. So you might have a bud that has more of is a naked bud that doesn't have these protective outer scales. This graphic illustrates how it looks as a breaking leaf bud changes into an actual leaf. So this walks you through the different stages. So on the left-hand side, first you have a bud that's enclosed in bud scales. The breaking leaf bud starts when you can start to see the leaf tips emerging from the end. And then you can eventually see the leaves unfolded on the upper right-hand side. And these leaves will be increasing in size until they reach their maximum size. And just to give you a little bit more information that relates to some of these phenophases, so there is the leaf blade here that you can see, and then that is attached to the stem of the plant by what is called a petiole or a leaf stalk. And that is a very important part of the breaking leaf bud phenophase definition. So this is the first of the phenophases that we'll be going through today. So the question is written on the data sheet as, do you see breaking leaf buds? And in the box here is one of the phenophase definitions. So this is the first time that you'll see this, and we'll have this throughout this presentation. Anytime you see this text enclosed in a box, that's actually one of our phenophase definitions that are available to you for all of our species protocols. So for breaking leaf buds, the definition is one or more breaking leaf buds are visible on the plant. A leaf bud is considered breaking once a green leaf tip is visible at the end of the bud, but before the first leaf from the bud has unfolded to expose the leaf stalk or leaf base. So in the photograph on the bottom left, first you can see that there's a bud, but there are no leaf tips visible. So you would report a no here for breaking leaf buds. In the next photograph, you can see that the green leaf tips are visible at the end of the bud. So you would report a yes for breaking leaf buds. In this last photo, you would report a no for breaking leaf buds, and that's because you can actually see that leaf stalk or petiole that's visible in between the leaf and the stem. So that would be a no for this question of breaking leaf buds. And not all of the buds on a plant will break at the same rate. So although some of the buds aren't breaking anymore, you should still continue to report the presence of breaking leaf buds until all of the buds have at least one leaf unfolded. So when you would no longer consider, or when you start reporting a no to breaking leaf buds, that would be when all of the buds on the plant have at least one leaf stalk or petiole that's visible. So here's the first of our quizzes that we'll be giving you today. So this question is, do you see breaking leaf buds, for the photograph here? And we'll give you a chance to look at this photograph for a few moments before we launch our poll. And we posted the definition again, so you can review that if you need to. But just take your best guess and try to decide whether or not you see breaking leaf buds. And you'll have either a yes, no, or a not sure option for this. So we'll let the answers come in for a few more moments. Okay, so it looks like 91% of you said yes. Just a couple people said no, and a couple people said not sure. So we would say yes for this question. So you can see that there might be a couple of these buds in this picture. For example, I would say the bottom one for sure. You can see that leaf stalk. So for this bud, you would say no for breaking leaf buds, and perhaps for this bud as well. But because there is still this one bud that's visible that has breaking leaf buds where you can see the green leaf tips, but you can't see the leaf stalk, you would report a yes. So for the overall question, you would report yes to breaking leaf buds. Okay, moving on to form phenophases. These are a bit different than the deciduous phenophases. So the first phenophase that you would record, initial growth, that kind of captures the same information as breaking leaf buds, where it's that first kind of spurt of growth that happens at the beginning of the season. And that you would report and up until when you started to see leaves on the plant. And then once you started to see leaves that were unfolded, you could report both initial growth and leaves until the end of the initial growth phase. And then once all of the leaves were unfolded, you would just report leaves for the rest of the season. And then for the flowers and the fruits, these are the same as for the deciduous tree phenophases. So to go over the definition for initial growth, it's the new growth of the plant that's visible after a period of no growth, such as in winter or drought, either as new green shoots sprouting from nodes on existing stems, or new green shoots breaking through the soil surface. For each shoot, growth is considered initial until the first leaf has unfolded. So you can see for this May apple on the left-hand side, you would report a yes for initial growth, because you can see this new growth visible, but the leaves are not yet unfolded. But for the picture on the right, you can see that the leaves are visible, they're unfolded, and so you would report a no for initial growth. And at this time, you would start reporting a yes for leaves as well. So you would report a no for initial growth, but you would be reporting a yes for leaves for the photo on the right. And moving on to leaves. So this is our definition for leaves. One or more live unfolded leaves are visible on the plant. A leaf is considered unfolded once its entire length has emerged from the breaking bud so that the leaf stalk or leaf base is visible at its point of attachment to the stem, but do not include fully dried or dead leaves. So if you see an example such as the photo here on this slide, where all of the leaves have dried and lost all of their color, you would no longer count this as leaves. You would report a no, as they're dead leaves at this point. Moving on to colored leaves, the definition for this is one or more leaves have turned to their late season colors, do not include fully dried or dead leaves that remain on the plant. So for colored leaves, you would report a yes, such as for this example here, and it doesn't matter why. So you might report this for the typical late season colors that appear in the fall, but you can also report leaves, for example, for summer drought or insect damage. As long as they're turning those late season typical fall colors, it doesn't matter the reason. Although if you do think that the leaves are changing due to either summer drought or insect damage or other stresses, we recommend that you put this in the comments for that observation. So if you see this color change happening at a different time of year than you would normally expect, say in the summer, you might report that you think that there's another reason that's causing this leaf color change, and just put that in the comments field. So here's your next quiz. So let's say in this example, you walk up to your tree that you're observing, and you see just a few of these colored leaves, but the rest of the plant is green. And let's say that this is not happening during the typical fall colors change time of year, but it's happening in the summer. So just take a moment to study this picture and try to decide whether you would report a yes, a no, or an uncertain. Okay, looks like most of you have voted, so we'll post the results. So almost everyone said yes, and we had, again, a few no's and not sure's. So we would say yes for colored leaves. So as we said, it doesn't matter the reason. So even if this is because this is happening in the summer and it's just a few of the leaves that are colored, you would still report a yes, but you should report a comment as well. For example, just a few leaves due to stress. Okay, so we're moving on from leaves to flowers now. And just to give you a little bit of background on flowers, this picture or diagram here shows a basic flower or a regular flower. And there's quite a bit of variation in flowers, so they won't all look exactly like this. But just to run through the basic anatomy of a general or basic flower, they normally have 4 different arrangements of flower parts. So there'll be sepals, which you can see here in the green, which are protective parts for the flower bud. Then the petals, which are the showy parts of the flower, those serve to attract pollinators. And then there are the male and the female reproductive parts. So the male parts here, the stamen, consist of the anther and the filaments. And then the female parts consist of the stigma, the style, and the ovary, and that forms the pistil. So some flowers are monoecious, so you would have flowers that actually have male and female parts which are not located in the same flower but are actually located on different flowers on the same plant. And you can also have dioecious plants where the actual male and female flowers are on totally separate plants. So if you end up with a male plant, you might never see fruits form, for example. And there are many types of flower structures as well, so they won't all look like that general or regular flower from the previous photograph. Sometimes you can have them structured in inflorescences. And so if you have a grouping of flowers, and this shows some of the different types of groupings here, even if you just see one flower bud or one open flower, you'd still report a yes for that phenophase, even if it's just one from that whole inflorescence. The definition for flowers or flower buds is one or more fresh, open, or unopened flowers or flower buds that are visible on the plant. Include flower buds that are still developing, but do not include wilted or dried flowers. So for these photographs here, you would report a yes for flowers or flower buds for the first picture, and you would also report a yes once the flower is open for the second photo. For open flowers, this is when one or more open fresh flowers are visible on the plant. They're considered open when the reproductive parts, the male stamens or the female pistils, are visible between or within unfolded or open flower parts. And again, do not include wilted or dried flowers. So for this example, for this Phenophase question, for the first picture, you would report a yes to flowers or flower buds, but a no to open flowers. And then for the second picture, you would report a yes for both flowers or flower buds and open flowers, keeping in mind that that flowers or flower buds is one of those overlapping phenophases that you continue to report. And just to emphasize that last part of the phenophase definition, do not include wilted or dried flowers. For some species, these dried flowers will persist on the plant for a long period of time. So in this example here for this oak, you would report a yes for the picture here because these flowers are still alive. But once they dry, even if they remain on the plant for a long period of time, you would report a no once they've wilted and dried. Okay, so the quiz for flowers or flower buds or open flowers. So we're asking for both of these here. So take a look at this picture and try to determine if you would say a yes or a no for either of these phenophases. So would you report a yes for just one of these phenophases, or a yes for both, or would you report a no for one or both of these phenophases? So we'll give you a moment. And you can go ahead and read those definitions again. I know there's a lot of text on there, so we'll give you a moment. Okay, we'll launch the poll. Okay, looks like people are still voting. We'll give it a couple more moments. Okay, looks like most people have voted. Okay, so most people said yes to both flowers and flower buds and open flowers. And yes, that's exactly what we would say as well. So I hope that looks like a lot of you got that take-home message that the flowers or flower buds is one of those overlapping phenophases. So you would report a yes to both of those. So we often get this question, how do I tell a leaf bud from a flower bud? And it really depends on the species that you have. So we really recommend, before you're really comfortable with a lot of these phenophases, you don't have to answer everything. So it might take you a year to see all of the phenophases occur before you can really know how to start reporting that phenophase at the beginning of the season. So you don't need to feel like you have to make an answer for everything right when you first start observing. You're welcome to put a question mark if you want to just write something down or just not answer it at all. So we just wanted to run through one example for this sugar maple of what it might look like. So at the beginning of the season, you might have a stem that has a few different buds on it, but you're not sure if it's a leaf bud or a flower bud. And sometimes, for some species, these leaves and flower buds might even develop in the same bud. So that's all the more reason to kind of wait and see what's going on with your species and get familiar with it before you start reporting. Oh, and I guess this is a red maple, not a sugar maple in this photo, so that's incorrect. So before you start reporting breaking leaf buds, if you remember that definition, you won't actually report a yes for breaking leaf buds until you see that green leaf tip visible. So you don't have to worry about reporting the breaking leaf buds at all until you actually see that, that open. And at that point, you'd be able to tell whether it's a leaf bud or a flower bud. However, for the flower buds, it's a little bit trickier because you actually start reporting on those before it opens and then before you really know what it is. So if we wait until later in the season and we actually see this open, we can see that the leaf was actually coming out of this top bud, the one that's the terminal bud, whereas the flowers are coming out of the buds that are the lateral buds on the stem. So these are the same two here in these two photos. So what you can do is, if you're unsure about what the bud is going to turn into, you definitely won't be reporting breaking leaf buds until actually you can see that green leaf tip. But you can always circle that uncertain option for flower buds and make a comment about it on that first date that you see the bud expanding. If it turns out to be a flower bud, then you can go back and report a yes to flowers and change that question mark to a yes for flowers or flower buds, starting with the date that you made that comment. You can see here, this is the lateral bud that turned into a flower there. So when a flower becomes a fruit, this is the last of the three categories that we'll be talking about today. So it is tricky to know when a flower first becomes a fruit. And we really don't want you to stress too much about this. We're not as interested in knowing when a flower first turns into a fruit, when it's that really tiny fruit that's just developing. We're more interested in when it's obviously a fruit. So you can always wait to report until you're sure that a fruit has actually developed, or you can use that question mark option. And keep in mind that flowers don't always turn into fruits. So if it's not pollinated, or perhaps if it's pollinated but not fertilized, or if the fruit is aborted on the plant, you might not always see a fruit. So don't be too worried if you never see that, even though you do see flowers on your plant. And then also, if you have male plants with male flowers, you won't have fruit. So keep that in mind and look at those species definitions to see more details about your particular species. And there are a great variety of different types of fruits. So this, again, is a great reason to study those phenophase definitions and refer to them as you're making your observations. So this shows just a few examples of the variety of types of fruits there are. Looking at the first example here for red maple, we've listed the definitions for both fruit and ripe fruit. So this tells you very specific information for the species. So for Acer rubrum, the fruit is 2 joined seeds in a V shape, each seed having a wing that changes from green or red to tan or brownish and drops from the plant. And it tells you specifically that ripe fruits is when it has turned to that tan or brownish color and readily drops from the plant when touched. So this really gives you explicit directions on when you would report fruits and then when you'd start also reporting ripe fruits. So you, for this picture, report a yes for fruit, but a no for ripe fruit because it's still very green in color and hasn't turned that tan or brownish color yet. Another example for this oak here, we have the fruit is a nut or acorn partially covered with a cap that changes from a green or a green-brown to brown, red-brown, or dark brown. And so that's telling you in the ripe fruit definition that it's considered ripe when it has turned that brown, red-brown, or dark brown color. So this one might be kind of borderline, but it looks still like it's mostly green. So you would report a yes for fruit and a no for ripe fruit. So here's another quiz for whether you see fruit or ripe fruit. And again, we're asking for both of those phenophases. So go ahead and read the definition for black cherry and decide whether you would report a yes for both of these phenophases or just one or a no for both. And we'll give you a couple more moments to read through those definitions. Okay, it looks like most people have voted, so we can take a look at the results. And it looks like most people said yes to both, but there were quite a few that said yes to ripe fruit only. So we would say yes to both. So again, the fruit phenophase is that overlapping phenophase. So even when you report a ripe fruit, you want to also report a fruit as well. So you would report, anytime you would have a ripe fruit, you would also report a yes for fruit. So just to go into a little bit more detail about some particular species that actually have a very quick ripe fruit phenophase that occurs. So we've actually created another phenophase that I'm going to talk about next to help with this. But in this situation, you might see that the ripe fruit for this witch hazel is when a fruit is considered ripe when it has turned brown and split open to expose the seeds. However, do not include empty capsules that have already dropped all of their seeds. So in the picture on the left, you would report a yes for fruit, but a no for ripe fruit because it is not yet split open to expose the seeds. In the middle picture, you would report a yes for fruit and for ripe fruit. But for the last one, you can see that it's actually empty inside. So the seeds or the fruits have dropped out of the capsule at that point. So you actually would report a no for fruit and a no for ripe fruit. So it might actually require, based on the species you have, really getting up close to the fruit and looking inside and just seeing if there's anything left in there that actually counts as a fruit. And due to this kind of species that we have on our list, we've actually created this phenophase that is, do you see recent fruit drop? And that allows you, in the case of this plant here, if first you went and you reported, say, for the photo on the left, and then the next time you went out to observe, you saw something like the photo on the very right, and you kind of missed that ripe fruit stage, you wouldn't be able to report ripe fruit for that species. So having this recent fruit drop allows you to report that and actually know that, yes, there were fruits and ripe fruits that dropped out, we just didn't get to see them. So the definition for recent fruit drop is one or more mature fruits or seeds that have dropped or been removed from the plant since your last visit. Do not include, obviously, immature fruits that have dropped before ripening, such as in heavy rain or wind, or empty fruits that had long ago dropped off all of their seeds but remained on the plant. So you can report a yes for this phenophase if there's a lot of unripe fruit from your last visit appears to have ripened and fallen, and you can actually see that on the ground. And you can also report a yes if a storm seems to have brought down a lot of ripe or almost ripe fruit. So if it's unripe fruit that the storm has brought down, you wouldn't report that. But if it seems to have brought a lot of ripe or almost ripe fruit down, then you would report a yes. And you can always use that question mark as well. If you can't figure out how long the fruit's been on the ground, if it was recent, you're welcome to use that question mark. So that is the end of the phenophase section. And I wanted to go into a little bit of information about intensity. And we won't be covering too much here. We actually had a separate webinar last year that you'll be able to view if you want as a recording, if you want more information on this. But just to go over a little bit of information about intensity. So if you answer yes or an uncertain to a phenophase, you can also report an additional question about the degree to which the phenophase is expressed. So this could happen, or this could, examples of this are how many leaf buds are breaking, what % of flowers are open, and how much pollen do you see. And these are kind of 3 different types of these intensity questions. So for how many leaf buds are breaking, you're going to be answering in more of a count kind of answer. And you can see on the graphic here at the bottom, the breaking leaf buds is this type of question where you would answer in a bin of numbers. So you can estimate whether it's a very small number, such as less than 3, up to a lot of these breaking leaf buds. There are also percentage questions, so we're asking things like what % of the flowers do you see are open? And then also more qualitative kind of questions, such as this on pollen, which you actually would answer as a little, some, or a lot. And when you answer these intensity questions, keep in mind that we don't really need 100% certainty. So we're really looking for a ballpark answer, estimating in one of those bins. So for example, in this picture, would you estimate the number of people in this room to 10, 100, or 1,000? So it might be a little bit more tricky than that if you're looking at a plant with thousands of breaking leaf buds, but just keep in mind that it's really just the approximate answer. We're just trying to get an estimate. So don't worry too much about trying to get the exact number. We also wanted to remind you that we're really interested in the change in these values over time, not necessarily the exact value. So one way to think about this for leaves, if we're asking about the % of the canopy that's full, what we're really interested in is how that's changing over time and when that peak is occurring. So you might start reporting in March, and as it generally increases and then decreases at the end of the year. And that's a lot more valuable to us than knowing exactly whether it was 25% or 50% at the start or when you first started looking at this phenophase. And just a couple tips for estimating intensity. In the webinar that I'll give you the link to in a moment, it describes this in a lot more detail. But for a lot of these percentage bin questions, you really have to know what 100% looks like for your particular plant. So again, you might need to have observed for at least a year in order to know what 100% really looks like. So for example, in the leaf canopy question, you have to know what your tree, the actual individual tree, looks like when it's at 100% canopy. So this would require seeing that tree at the time of the year when it was the most full of leaves. So when you're answering these intensity questions, you'll want to picture the tree, what it looks like at 100% canopy, and then visualize if you're looking at it when it's not quite at 100%, you can imagine drawing like lines over it to separate it into a few different quadrants, and then trying to visually maybe move these leaves and see if you could fill up, say, 50% or 75%, 2 or 3 of those different quadrants, to tell how much of that potential canopy is actually full with leaves at that time. So you might answer for this picture on the right that you could visually imagine kind of moving those leaves into maybe 2 or 3 of the quadrants, so you would get 50 to 74% leaf canopy cover. For estimating intensity for these count kind of questions, such as how many breaking leaf buds do you see, or how many flowers or flower buds do you see, you might estimate what a particular section on the tree looks like. Say, how many flowers do I see in this section? And you could stand a little bit back from the tree and hold up your fist or hold up a clipboard and try to visualize how many flowers or breaking leaf buds are behind that space, and then count how many of those spaces there are on the tree, and kind of do a multiplication to figure out how many, approximately how many there are on the tree. So then you would kind of line up those spaces and try to figure out the coverage. So if you want more of this kind of information, tips on estimating intensity and more on the different types of intensity questions, you can check out our recorded webinar. I think Sarah is going to post this link in the chat box, and that's on our webinar archive. The video quality isn't the best on that recording, so if you need a better quality, you're welcome to send an email to one of us, or you can email that meeting's email address where you got the reminder, and we can send you the actual video file. So I wanted to go through a lot of the resources that we have available for you in answering these kind of phenophase questions. So the number one place to start is the phenophase definitions, because these have the species-specific information that you need to answer each of the questions. And you can find these on a few different places. So on your observation deck at the bottom, any plant that you add to a site, you'll then see the species profile, which includes the definitions. And you can also print a phenophase definition sheet there as well. You can also find this information on our smartphone applications. So next to each of the phenophase questions, you'll see a little eye. And if you click on that on your phone, then it'll pop up with that phenophase definition for that species. We also have a beautiful new Botany primer guide. So this has a lot of information about the things that I've been talking about today, as well as a lot more detail. Here's a screen cap of the table of contents. And so there's detailed information about all of these different phenophase categories, including lots of photographs with graphics like arrows and circles that point out specific parts that you should pay attention to of the different structures. And then there's also a quiz in there, and references and lots of other resources. So Sarah's going to type in where you can download that. It's freely available for download, and we're also looking into options to have a printed version of this as well for purchase. But right now you can download it and print it out, and it's available on the link that Sarah sent out. Also, you can try the frequently asked questions page. This is located at the bottom of all of the Nature's Notebook pages. So if you just scroll down to the bottom, you'll see that little link for FAQ. And it takes you to this page, and it has all of the commonly asked questions that we've gotten. We update that every year. And so you can look first to see if your question's on there. If it's not, you're welcome to email observe@usa-npn.org, and we'll do our best to answer your question. I also recommend that you use your phenology calendar. So once you're a Nature's Notebook observer, you have a customized phenology calendar right on your observation deck. And this is a great way to kind of check over what you've entered for different species. So it could really help you figure out that maybe you entered a no and you should have entered a yes, or vice versa. And you can always go back in time and change your observations that you've already entered and correct them. If you're wondering about what species to observe, or you want to contribute to one of our campaigns, you can find out more about those on this address. Sarah will type in the URL as well. So we have different campaigns for cloned and common lilacs, cloned and native flowering dogwoods, and green wave species, including maples, oaks, and poplars. We also have a partner campaign, Pop Clock, where you can track balsam poplar and quaking aspen. And these are a great way to help researchers answer key questions. And when you sign up for these campaigns, you'll be getting emails every month or every 2 months that have results and actually show you the data that you're submitting, and then also include an end-of-season summary at the end of the year. So it's a great place to start if you're not sure what to observe or you want to really contribute to the science that's happening. And lastly, here is information about some of our upcoming webinars. You can view those on our webinar page. And we have another webinar coming up June 1st. That's the second part of our spring casting, which we've been doing for the last few months. Some of you might have joined us for our first webinar that was kind of introducing the idea of that effort. So this next webinar is going to be summarizing the results, looking at how we did on our predictions of when lilacs were leafing out during the spring. We've been sending out emails to observers around the country asking them to check on whether our models are correct. And so we've been getting reports back from them, and we'll let you know what the results look like for that. We also have a spring summary campaign coming up on the 16th, where you can see the results from some of your data that you've submitted. And then a webinar in September that's more for our local phenology leaders to tell you more about how some of the other Nature's Notebook groups are using Nature's Notebook. So with that, I wanted to post this quote, which came up in one of our past webinars, actually our intensity webinar, from one of our observers who said, "The best advice is don't stress, do your best." So just keep in mind that there's not always a right answer, and just do your best and don't take too much time to try to get the exact answer. You can always use those question marks, or just don't report things until you're comfortable with them. And so thank you for attending the webinar, and also thank you to all of our observers. This is a screen cap from our Nature's Notebook homepage just this morning. We're at 24% of our goal of 1.5 million records for this year. And you can see our tree is already filling up with lots of the different animals that represent the records for this year. So thank you for that. And now we'll have our question and answer period. You're welcome to stick around for that. We might go over our hour here, but we are recording the webinar, and we'll be reporting the link for that, as well as the question and answer document later on today. So we're going to check out and see the questions that have come in so far, and Ellen will be answering those for you today.
Okay, so the first question that came in, actually I think Erin covered this. The question came in just before she answered the question, but I'm going to reiterate. The question was for flower buds. What do you report if you see unopened buds? You cannot yet tell if they are leaf buds or flower buds. And as Erin had said, you might not know this until you get comfortable with your tree, your species. And oftentimes, buds on trees, flowers open first before leaf buds. And if you don't know that, you don't really know what you're looking at yet. And so you might need to give it a little bit of time before you're comfortable deciding whether they're leaves or flowers. And you can report question marks. If you see sort of swelling flower buds, or swelling buds, and you're not sure if they're leaves or the flowers, and you kind of want to be reporting that they're, flower buds that are starting to develop, just put a question mark for that until they actually open, and you can see the petals, and then you know that they were flower buds. Another question that came in canopy, would you include dead limbs as part of the 100% of the tree? And the answer to that is no, you would not, because what we really want to know is, given the full potential canopy of the tree, which might not look like a full green shape, a full green circle or semicircle. You just want those parts that you know are going to get full. And so we want to know, relative to that, are you at half of that, or 50% of that, or 75% of that, or what? So you can ignore those parts where you have a dead limb on a tree that never gets green when you're doing your canopy intensity measurements. Another question we have is, how do we track pollen, such as red maple? And I don't think the pollen, we didn't show the Phenophase for pollen on the screen, but basically the question is, do you see pollen? I don't have it in front of me here. And the answers are, the choices are, I see a little bit where there's just a few grains. I see some where it falls out on you're looking on a piece of paper or a dark surface, or lots. And in that case, it's often in some species, as in pine trees, you can actually see it flying from the tree. You don't even need to be up close, and that's lots of pollen. Again, it's a very qualitative answer, but that's all we're really looking for. Other people measure pollen more very quantitatively and with very complicated methods. How do you change your observations if doing this online? I think Sarah
when you can log into your observation deck, go to your observation deck, and in the far right-hand column where the Enter Observations link is, click that, and then using the blue arrows at the top of your screen, you can scroll back to the proper date or jump back a number of records. There's an option for that at the top. Say if you want to jump back 10 columns. So you go back to the date, and then you can select, change the yes, no, or question mark answer, and then submit at the bottom as you would have done for a new record.
There's also a frequently asked question about that. So if you want to look at that answer again that Sarah just said, you can go to that frequently asked questions page and review it there.
Okay, there was another question that came in that I wanted to just talk a little bit about. Somebody asked if there are pictorial guides to Phenophases available, and we are working on what we're calling a Phenophase primer that is the sister to the Botany primer, and that will have much more explicit information about how to interpret all the Phenophases for our different functional groups. So that would be forbs and grasses and deciduous trees and shrubs, evergreen trees and shrubs, semi-deciduous trees and shrubs, etc. And that it will not be an exhaustive document that has photos of all of the species on our list, but hopefully enough that you kind of get an idea of the range. Hopefully there'll be pictures in there for a species that's quite similar to the ones that you're observing that will help you in much more detail kind of feel comfortable evaluating the Phenophases. And that is due out in the spring of 2016, so in about a year we'll have that. Somebody's asked a question, if we're not sure of what we're seeing, can we send a photo for help? And we do have a botanist on staff, and we only have one, so we don't want to overwhelm her. However, often we can help you, but the photo needs to be really very good photo and often a close-up of the plant structure, so keep that in mind. How often, another question, how often, daily, weekly, would you like observations? And what we generally say is once a week would be great if you can manage that as a general rule. However, there are times a year when we don't want you to, we don't want to overwhelm people or make them do busy work. There are times of year when it doesn't even make sense to do any observation. So I live in Maine, and kind of all winter long in Maine, if I'm only doing plants, everything is dormant. So I kind of don't even bother to go out for about 3 months in the wintertime. But then in the spring, when things start to happen really quickly, I try to go out every 2 or 3 days to capture all the very quickly changing plant stages. And so it depends, it really depends on the time of year, but in general, once a week, more quickly or more frequently when things are changing quickly. Okay, next question. On initial growth, if you have a patch of trout lily and some are breaking through, don't understand why we say no if they are starting their initial growth. Okay. Not quite sure. Let me think about this for a sec.
How about we get back to you offline? We'll email you directly with an answer for that question because we are out of time. And that's all the questions we've got. So again, if you ever have a question, feel free to email us. Hopefully we will see you in a future webinar. Happy observing!